All right, so this is part 2 in our Excitable Cells Chapter 8 lecture. And so here we're going to begin looking at different types of ion channels. So if you recall from the first part, and if you haven't watched the first part, go watch the first part, that we said that the movement of ions that we're going to talk about in our action potential and that contributes to our resting membrane potential and whatnot, this movement was a type of diffusion, right? So diffusion was a type of passive transport. And we said specifically that there were two types of diffusion. There was simple diffusion and there was facilitated diffusion, right? So we said that it turns out that our movement of ions for our action potential occurs via facilitated diffusion, and facilitated diffusion means that we have a carrier protein or transport protein that assists in the movement of that ion, right? So instead of just diffusing directly across the membrane, it has to diffuse across the membrane via some type of channel, right? And so we have two general types of ion channels that we'll look at, and we'll focus on one in particular, and then as we move into future lectures, we'll discuss the other type, all right? So both of these, all right, so both of these ion channels, in general, they both have similar features. One, they both allow the movement of an ion to go down the concentration gradient, right? So in other words, they allow the movement of ions to go from high concentration to low concentration. So if you think of potassium, for example, potassium is highly concentrated on the inside of the cell. So whenever one of these channels opens either voltage gated or ligand gated, they both allow potassium to move out of the cell, all right, if they were a potassium channel, all right? So that's another thing, is that these channels, in order to allow an ion to move, it has to be specific to that ion, right? So sodium only moves through sodium channels. Potassium only moves through potassium channels. Calcium only moves through calcium channels, and so on, okay? So we're moving from high concentration to low concentration. They both are ion specific. Now, they differ in terms of how they open, all right, or what they respond to, all right? And this will be the main difference between these two, all right? So the first one we'll talk about are voltage-gated ion channels, all right? So voltage-gated ion channels, these allow ions to move from high concentration to low concentration, down their concentration gradient, and they open in response to a change in the membrane potential, all right? So in other words, a change in the voltage or a change in the electrical charge across the membrane, okay? So we said that we have lots of sodium on the outside, relatively little sodium on the inside, lots of potassium on the inside, relatively little on the outside, and you have lots of other things. All of these things, molecules and ions and compounds, they all have a charge, right? They're either positive or negative, right? Or they could be neutral, I suppose. So depending on the distribution of these different ions and molecules and their charges, you're going to get a difference in the charge across the membrane, all right? And this is known as the resting membrane potential, right? And it turns out that the resting membrane potential, we'll talk about this more later, it is negative on the inside relative to the outside, okay? So for our neuron, for our nerve cell, it's somewhere around negative 70 or so, okay? Negative 70, negative 71 or 2 or something like that, all right? So all this to say, the difference across the membrane the membrane is polarized. So in other words, there's a difference in the membrane charge on one side versus the other side. Any stimulus or anything that causes that potential, that membrane voltage difference to change, could potentially cause these voltage-gated ion channels to open, right? And each individual voltage-gated channel, whether it's sodium or potassium or calcium, opens in response to a different change in the membrane, right? So it opens in response to a different voltage. So sodium channels open at a different membrane potential or a different charge than potassium channels, and those open at a different membrane potential or voltage than calcium channels, and so on and so forth, right? So if you remember from if you remember from chapter 2, when we discussed the idea of protein synthesis, right? We talked about we have a DNA and you transcribe the DNA into RNA, and then you take your RNA and you translate it into a protein, and you take your amino acids that are in the cytoplasm and you bring them together with tRNA to the ribosome, and you get a nice protein, right? Well, it is the amino acids, or these kind of end regions of the amino acid that we're calling an amino acid residue in this protein on either side. it's the amino acids that sense the change in the membrane potential, right? They sense the change, or the change in the charge, the change in the electrical charge across the membrane, across the plasma membrane. And when it does so, if that change in the membrane potential, if it is appropriate, or if it is the correct change for that particular channel, it will cause that channel to open, right? And so we said in chapter 2 that a protein gets its function based on its shape. And so depending on the shape and the formation of a particular protein, you're going to get a different function. And so whenever the protein channel changes shape, or in other words, it opens, it's going to change the function of that protein. So it's either going to allow things to move through it, or it's going to block the movement of things through it, right? And so here you can see we're very, very positive on the outside, very, very negative on the inside. And then we become relatively less positive and relatively less negative here. And that difference in the charge results in a change in the shape of the protein. And so you get the protein going from this closed conformation, this closed state, to this open state. And now if this was a sodium channel, you'd have sodium ions on the outside and they would flow in, and then ultimately the charge across the membrane would change and this membrane channel would close, and then you'd no longer have the movement of ions. All right. So if we look at our second type, we look at ligand-gated channels. So voltage-gated, they were gated by the voltage. They opened in response to a change in voltage. Ligand-gated, these are membrane channels that open in response to a ligand, right? And so all a ligand is some type of molecule. It's some type of chemical messenger that has a binding site on this channel. And so this channel it serves two functions. One, it serves as a receptor, okay, because it has to bind the ligand, so it's a receptor. And then after it binds the ligand, it opens and it becomes a channel, right? So it's really a receptor and it's a channel. One example of a ligand that we'll discuss is something called acetylcholine. This is a very important neurotransmitter that we'll discuss some more in chapter 9, so the next lecture, as well as later in the semester when we get towards chapter 14 and 15 when we talk about the autonomic nervous system. But acetylcholine is the main neurotransmitter that we'll discuss that's released from our neuron, right? So we have an action potential that we'll get to. The way you relay the action potential from one cell to the next is via acetylcholine. All right. Now there's other types of neurotransmitters as well. They don't all release acetylcholine, but acetylcholine is going to be the main one that we discuss. Right. So it binds to the channel. In this case, it's acting as a receptor. When it binds as a receptor, it opens, it becomes a channel, and again allows things to move in. All right. So the way that these ions are going to move, so we said that they can either move through a voltage-gated channel, they can move through a ligand-gated channel. In order for them to actually move, however, there are two forces, right? One of them is the concentration, and one of them is the charge, or the electrical charge across the membrane, okay? So we've already discussed a little bit of these, but there's also an interplay between the two, right? So we said that potassium, if we take the example here, potassium is very highly concentrated on the inside of the cell, right? So we said it's very highly concentrated on the inside, not very concentrated on the outside. And so potassium has a tendency to go down its concentration gradient, and that would take potassium outside of the cell, right? If we were talking about sodium, we would be moving inside the cell, right? So if we look here we're inside the cell, intracellular, lots of purple potassium here, and it will move out, and you have very little, relatively little potassium on the outside of the cell. All right, so that's one force. You're moving from high concentration to low concentration. That random Brownian movement causes things to move down their concentration gradient. However, we have a second one as well, and this is the electrical force, or the charge, right? So if you have lots of potassium here, this potassium is very positively charged, right? And we have relatively little positive charge on this side, okay? So if we're moving potassium in one direction or the other, potassium itself is positive, and as you move more potassium to one area, that side or that region becomes more positive. And so as that region becomes more positive, it's going to repel the additional movement of more positively charged potassium ions, right? So you have what's known as this electrochemical equilibrium, right? So you have the force of potassium ions moving down their concentration gradient is balanced by the electrostatic repulsion of the positively charged ion, as well as the chemical movement or the concentration gradient as well, all right? So you have this electrical force, you have this chemical force, where these two meet, and when we're balanced, that's known as the electrochemical equilibrium, and the voltage across across the membrane for that particular ion, that's known as the equilibrium potential for that ion. So potassium has its own, sodium has its own, calcium, chloride, magnesium, so on and so forth. All right. We'll focus primarily on sodium's and potassium's equilibrium potential here in just a little bit. Okay, so if we review here, we have three main factors, right, that influence how our ions move. You can have ions moving from high concentration to low concentration, right, down a concentration gradient or up a concentration gradient, from high to low and low to high. You have the electrical driving force as well. So like charges tend to repel each other. So sodium is going to be repelled by other positively charged ions, and it would be attracted to negatively charged ions. And then lastly, the one that we haven't mentioned directly, but we've mentioned it kind of as we mentioned, the idea of ion channels, is that the membrane has to be permeable to a particular ion, right? So sodium and potassium are unable to move through the membrane themselves because they themselves are not permeable, right? So these are positively charged ions, and then you have the phospholipids of the membrane, and the two do not mix. The two are not permeable. So in other words, you have to have some other means of getting sodium in and potassium out, right? And so the way you do this is you have some type of ion channel, voltage gated or ligand gated, that allows the movement of an ion to go from one side of the cell to the other down its concentration gradient. Okay, so we'll also talk about later on ions moving up their concentration gradient or against their concentration gradient, but by and large the main movement or the main type of diffusion we'll talk about will be this idea of moving from high concentration to low concentration. Okay, all right, we're going to stop here, and we'll pick up by talking about specific types of ions or different types of ions in our next part.